We are going to start with a subunit regeneration. Many fully differentiated organisms have the ability to give rise to new individual organisms from their body parts. Means some of the organisms have the differentiating capacity or regeneration capacity. From that, their body parts are going to develop. Okay, that is, if the individual is somehow cut or broken up into many pieces, many of these pieces grow into separate individuals. See, example, if I take XYZ organism and I just mince it, I just made small pieces of that. No doubt I have cut it in a smaller pieces, but that smaller pieces are going to develop into a new individual. For example, simple animals like hydra and planaria. So we have example here, hydra as well as planaria can be cut into any number of pieces, and each piece grows into a complete organism. As I said, if the organism is cut in a small pieces, then that individual piece is going to generate new one. This is known as regeneration. See, this capacity is known as what? Regeneration capacity. Next, regeneration is carried out by specialized cell. See, for the regeneration ability, they have specific cells. These cells proliferate and make large number of cells. See, means what? Every individual cell, tissue and organ have that proliferating ability, means regenerating ability. From this mass of cells, different cells undergo changes to become various cell types and tissues. See, as I said, each and every mast cell having that capacity, okay, to regenerate that particular organ, tissue or cell. This takes place in an organized sequence referred to as development. See, if I cut this is the planaria. If I cut this in three sections, then the head section is going to develop the remaining body part. Whereas if I go through this middle piece, then it is going to generate head and tail regions and new individual. After that, the tail region is going to develop the front part or the head part. Okay, see, however, regeneration is not the same as reproduction. See, we cannot say this is the reproduction, since most organisms would not normally depend on being cut up to be able to reproduce. See, sometimes accidentally, intentionally, it is going to happen and they need to regenerate. Okay, next is what? The budding process. Organisms such as hydra, see, this is the hydra, use regenerative cells for reproduction in the process of budding. See, in hydra, a bud develops as an outgrowth due to repeated cell division at one specific site. These buds develop into tiny individuals when they get mature, when they are fully mature, detach from the parent body and become new independent individual. See, means that when the hydra is matured, it is going to accumulate all the cells on a particular part. You can say a bump here. Okay, after that it is going to push that bud, that it can grow into a new. Unless it is mature, it is not going to detach. You can see now here, it is completely mature one. After that, it is going to detach from the mother body. Okay. Next, vegetative propagation. There are many plants in which parts like the root, stem, and leaves develop into new plants under appropriate condition. See, if I'm taking pothos or the money plant as an example, only when we are taking one node along with a root, one node along with one leaf and one root. This is more than enough to grow in a new vine, pothos vine. Okay, pothos plant. Next, unlike, see, even though only this much part is present, then see what we need to do. If I'm propagating this in a water or else I'm propagating this in soil, no doubt I have sowed it in the soil or I have kept it in a water. But sometimes if conditions are unfavorable, at that time it is going to start rotting. The decaying process or the decomposition process is going to begin. But if the favorable conditions are present at that time, the small leaves are going to get out of here from the nodes. Okay, and it is going to develop into a new individual. See, unlike in most animals, plants can indeed use such as mode of reproduction. See, we can say this is as one of the type of reproduction. This property of vegetative propagation is used in methods such as layering or grafting to grow many plants like example is sugarcane, roses or grapes for agricultural purposes. See, if I go through sugarcane first, the stem is present, right? Along with that, some nodes are also present, some internodes are also present. Okay, what they are going to do, along with this stem, some aerial roots are also present in that nodes. Okay, what the farmers do, they are going to cut in the internode. They are going to cut it along the internode, and now this segments or the parts having a two individual. How I'm telling two, it can be a four. Why not four? See, here also roots are present, here also the roots are present. Okay, but sometimes they are doing only two or sometimes they are doing it as a four. Next, coming to the rose, what we are doing, we are just bringing the one stem from our friend, neighbor or someone. Okay, and we are just going to sow it in a we are just going to sow it in soil. So what is going to happen after that? See, here the roots are going to develop, but we need to provide the favorable condition on it. Next, coming to the grapes, it has a particular vine which need the support, and if we are doing for the agricultural purpose means we can say it is a commercial base. We are sowing and all this, after that we are getting the benefit from that. Plants raised by vegetative propagation can bear flowers and fruits earlier than those produced from seeds. See why this is happening. Firstly, we have drafted it or we have cutted it, we have propagating it from the fully mature plant. That's why the matured cells are present in this one as compared to the plants produced from the seeds. Okay, such methods also make possible the propagation of plants such as banana. Example, we can also propagate to banana like this with the help of this method. Orange, rose, jasmine, that have lost lost the capacity to produce seeds. See, if they are unable to produce the seeds, then also we can propagate them. Another genetically similar sorry another advantage of vegetative propagation is that all plants produced are genetically similar enough to the parent plant to have all its characteristics. See, if we are doing with the method of this vegetative propagation, we can say they are identical to their parents, genetically identical to their parents. How I am telling this? Because the genetic content will be same in their body. Okay, see, we have one of the activity. Take a potato and observe its structure. Can no notches be seen? See, cut the potato into small pieces such that some pieces contain a notch or bud or eye and some do not. Spread some cotton on a tray and wet it. Place the potato pieces on this cotton. Note where the pieces with the birds are placed. Observe the changes taking place in these potatoes pieces over the next few days. Make sure that the cotton is kept moistened, which are the potato pieces that give rise to fresh green shoots and roots. See, what we need to do, we have one of the potato here. We have one potato, assume that it is a potato, and it having some of the notches or the eyes, particularly it is known as what, eyes. Okay, so what is going to happen, we are just making it in a pieces. That potato is cut into a pieces, but some of these pieces having the notches and some don't have. So what we are going to do, we are just placing it on a cotton tray. See, a tray is filled with cotton. A tray is filled with cotton. We are going to place the cut pieces. We are going to place the cut pieces. On some notches are present, on some notches are absent. See, after few days, what is going to happen? The notches are going to develop into, they are going to develop some of the shoots and the roots. They are going to develop some of the, see, wherever the notches are present, they have developed, and some roots are also present. So this is going to happen in this activity. So this show that they having some of the birds present on their body. Similarly, buds produced in the notches along the leaf margin of bryophyllum fall on the soil and develop into new plants. See, this is what example of bryophyllum, in which the leaf is only responsible for the production of new plants. Okay, so this is nothing but with the process of budding itself. So next we have activity, select a money plant. As I have said you, Cut some pieces such that they contain at least one leaf. Cut out some other portion between two leaves. See, dip one end of all the pieces in water and observe over the next few days which one grows and give rise to fresh leaves. What can you conclude from your observation? As per this activity, as I said in the money plant example, so here when we are going to dip this in water, so it is going to start developing the roots and the shoots part. Okay, same. Next, we have tissue culture. In tissue culture, new plants are grown by removing tissue or separating cells from the growing tip of a plant. The cells are then placed in an artificial medium where they divide rapidly to form a small groups of cell or callus. The callus is transferred to another medium containing hormones for growth and differentiation. The, the plantlets are then placed in the soil so that they can grow into mature plants. Using tissue culture, many plants can be grown from one parent in disease free conditions. This technique is commonly used for ornamental plants. Okay, viewers, so tomorrow we'll meet at the same time. Inshallah, Allah Hafiz, दुआ में याद रखना.